Greetings. Today, we will be talking about a short analysis for testing pre-existing quantitative leaf data from a tropical dry forest in northwest Costa Rica and testing to find if that data can act as a predictor for relative successional stage. Buzzard 2015 gives us access to several tree and leaf functional trait measurements of plant species residing within defined plots in the Costa Rica Guanacaste Conservation Area. This data gives us access to age, plot, species, abundance, biomass, area, and many leaf measurements, most notably being leaf carbon to nitrogen ratio and stable nitrogen isotopes. This image shows the location of the conservation area World Heritage Site where these measurements were taken. Additionally, a cursory understanding of succession is necessary. Succession is the process by which a forest transforms from a barren land to a habitable forest. There are four main stages of primary succession. However, these stages look different and progress differently depending on many factors, including climate, plant species, soil quality, and several other factors. By establishing a baseline of relative plot succession stage by tree count and area coverage, we can establish which plot is in which relative successional stage. The relationship between fungal and bacterial communities in forest succession is an important one, and it is mediated through the amount of available resources these communities can provide to the soils of the developing forest. As you can see, there is a relationship in the amount of fungi and bacteria that is present at the different stages of succession. As the different stages occur, there is a gradual increase of fungi and an increase in bacteria, followed by a gradual decline relative to the amount of fungi. Fungi is much larger than bacteria in biomass and outcompetes bacteria for space, reducing the available nitrogen in soil from bacteria communities. The amount of fungi directly influences the soil's supply of available carbon, and therefore facilitates the growth of mature trees. This relationship between fungi to bacteria ratios is a complex association that has correlational trends to succession. Successional stages are therefore characterized as large tree growth for more available carbon in soil, and this available carbon is directly related to the increase of fungi. The more available carbon in the soil, the more in biomass these trees are able to produce. Therefore, biomass is a metric that should correlate to the progression of succession. Buzzard 2015 references from Hobby 2003 that stable nitrogen isotope leaf measurements are positively correlated with the relative amount of nitrogen fixing bacteria. As succession progresses, the number of bacteria do increase. However, that increase relative to the total area of biomass that plots with larger trees can produce does decline. Therefore, we should expect a logarithmic increase in leaf nitrogen through the successional stages. Our hypothesis can therefore be tested with the following. If stable carbon and nitrogen isotope leaf measurements are known to correlate to fungi and bacteria ratios, then there should be a statistically significant relationship between these leaf measurements and relative successional stages. In order to analyze this data and test this hypothesis, we can perform primary statistics of all leaf measurements. However, the two that we can expect to more strongly reflect their biotic soil communities, biomass as a reflection of available carbon in soil, an association represented by fungi and mature tree species. Additionally, however, leaf nitrogen isotopes measurements reflect bacterial associations. Performing a multiple linear regression analysis with biomass and nitrogen isotopes as a predictor, independent variables should result in the following predicted trends. The analysis of establishing successional stage, shown here in Figure 1, shows the increasing age of plots left undisturbed. This data has already been ordered by plot number to reflect this. This is sufficient in establishing our relative successional stages, in which plot number is in ascending order from youngest to oldest in 9 ages ranging from 10 to 120, with an N of at least 11, as made apparent by the histogram of plot frequency shown on the right. In this data set, total plot biomass is a positively correlational to the plot, further supporting the establishment of the trend expected to that of successional stages, expecting denser forests through time. Writing a simple program in RStudio allows us to provide a comprehensive and detailed list of any correlational association crossed between every possible variable combination, which then filters for any strong and weak correlations. Shown here in Table 1, these tables were generated with CRAN's GT packages. The testing for total plot biomass across plot found a statistically weak positive correlation, shown here in Figure 2. The testing for leaf nitrogen stable isotope measurements across plot was found to not have any statistical significant relationship, shown here in Figure 3. Additionally, however, there were other statistically significant correlations to either plot or age. However, this was for total plot basal area and average individual tree basal area. Linear regression of age across the plots represents successional stages in terms of age of forests left undisturbed. And these results were strongly positively correlated. The plots are already ordered. However, the data will be represented in bins of each of the 14 plots. This ascending order made it simple as we can define the age of every plot and assign it as an index in that order. This relationship then confirms the assumption within our hypothesis that the rest of the analysis can rely on, which is that these plots in order by age do represent the relative successional stage. Analysis of the total plot biomass within a given plot across each plot shows a weak positive correlation. Our assumptions rely on the total amount of biomass inside of a plot being representative of the total amount of carbon available in the soil, which is associated with fungal associations of soil carbon production. The results seem to support this assumption, as we can see a weak positive correlation, suggesting that as these plots age, they are being supplied with increasing amount of carbon, which facilitates their growth. Additionally, the correlations with age or plot to basal area and total basal area seems to support this as well, as these variables follow the same logic with facilitation of growth supplied by more carbon as the trees and the soils that facilitate their growth age. Unfortunately, there was no association between leaf nitrogen stable isotope measurements and relative succession stage. The assumption was that as succession continues, there would be an increase of nitrogen stable isotope leaf measurements, followed by a general plateau as bacteria communities grow and then are outcompeted by fungal communities. Additionally, research has shown that the leaf nitrogen stable isotope is strongly correlational to bacterial communities' proliferation, suggesting that some factors affecting the outcome of not having increasing stable isotope measurements. The goal of this analysis was to further establish previously researched ideas in both succession and bacterial fungal associations. There was a hope to establish a strong connection between the succession stages and the ability to predict fungal and bacterial associations through ecosystem features through those stages of succession. Unfortunately, we could not find a predictor variable for bacterial associations, the known variable being leaf stable isotope nitrogen as a known correlative. It would be then that there are other variables not measured that have some impact disrupting that known association between leaf measurements and bacteria, or that bacterial communities have a unique pattern through succession that is not standard in this environment, or that leaf measurements are simply not correlational to succession, regardless of known association with bacteria. Regardless, it would be of great use to acquire a predictor variable for relative stage of succession, as it would assist in identification for forest rehabilitation.